welcome everyone, and thank you for this kind introduction. I was very happy and honored to be presenting here today, so thank you for this opportunity. And now I would like to present my work that focused on the selection of aptamers of antiviral effect. So our target molecule, I can't seem to move my shares. Yes, now it's working. Okay, so our target was the respiratory syncytial virus that causes lower respiratory tract infections with a disease burden comparable to influenza. It is responsible for significant morbidity and mortality among young children and the immunocompromised. Its name derives from its characteristic cytopathologic effect, the formation of syncytia, that means the fusion of the membranes of the neighboring cells. As of today, there is no licensed vaccine available for the prevention of RSV infection. Only a prophylactic treatment can be administered to high-risk infants with the monoclonal antibody polivizumab. Detection of RSV infection relies on qPCR of the viral genome or rapid antigen tests. However, the high cost of qPCR and the fragility of antibodies in rapid antigen tests make these methods less suitable in resource-limited settings. To overcome this problem, our group previously selected aptamers of diagnostic potential for RSV, which were able to distinguish RSV from its most common co-infecting agent, the rhinovirus in throat swab matrix. RSV has three surface proteins. G is a highly glycosylated protein that is responsible for the attachment of the virus to the host cell. The F protein mediates the fusion of the two membranes, and there is a small hydrophobic protein that acts as a viroporin. Upon natural infection, both G and F protein are targeted by antibodies. However, F protein elicits higher amounts of neutralizing antibodies. This F protein undergoes a dramatic conformational change upon the fusion. Therefore, we distinguish a pre-fusion and a post-fusion conformation. Polivizumab, the prophylactic drug used, binds both of these conformations via site 2 that is labeled with yellow here. However, recently, more potent neutralizing antibodies have been found to target the pre-fusion F-specific site 0 that is shown in red here. These neutralizing antibodies stabilize this conformation and thereby inhibit viral infection. Theoretically, it is possible to achieve the same effect with aptamers, so that was our long-term goal, to provide antiviral aptamers that target the pre-fusion F protein. In order to realize our goals, we decided to use modified aptamers. A tryptophan-like side chain holding nucleotide was chosen to be incorporated into aptamers. With that, we could target the site 0 on the pre-fusion F protein that is rich in hydrophobic amino acids. This particular nucleotide was incorporated by Codexel enzyme into amplicons. However, this is a proofreading activity lacking enzyme, so we also analyzed these amplicons by high throughput sequencing and found that the substitution of a natural nucleotide to a modified one had only a marginal effect on the error rate of Codexel enzyme. So we set out to produce the aptamer library, during which we experienced an excessive byproduct formation. We could evade this problem by the introduction of emulsion PCR. In this water and oil emulsion PCR's water droplets, the PCR reaction can occur physically separated from each other, and this decreases the chance to produce byproducts. This aptamer library was also analyzed with high-throughput sequencing, and we found that the diversity of the aptamer library was unaffected by the substitution of a natural nucleotide to a modified one. Then we exploited this modified aptamer library in a ModSelex protocol, in which we targeted a stabilized version of the pre-fusion F protein, and we used the post-fusion conformation as a target for negative selection. We ensured the specificity of these aptamers by introducing various competitor molecules, as well as increasing the selection pressure each round. Finally, we cloned and Sanger sequenced the final oligonucleotide pool, and we performed in silico analysis as well. And we found that 10 aptamers were present in multiple copies in the final oligonucleotide pool, showing a definite enrichment of potential RSV F protein selective aptamers. However, based on our previous results, we found that the most abundant aptamers might not necessarily be the best binders, so we screened all these aptamers using AlphaScreen. This method is an alternative to ELISA, and it measures the interaction between biomolecules that are immobilized to beads. This method revealed that there were 14 aptamers that showed binding to the prefusion conformation of the F protein, and these were chosen for further analysis with the same method. Here we challenged these aptamers with a varying concentration of the F protein as well as the postfusion conformation of this protein. These aptamers were able to differentiate between the two conformations. At least a 2 times higher fluorescence value was measured in F protein, the prefusion F protein containing samples. Aptamers that excelled at distinguishing the two conformations were D4, F5, G6, and H9. So in order to determine the dissociation constants of these aptamers, we chose yet another method called microscale thermophoresis. This method measures the interaction between two partners in free solution, and we measured that the affinity of our aptamers towards the prefusion conformation of the protein was at least an order of magnitude higher than compared to the postfusion F protein. This further validates our previous finding with the AlphaScreen. However, interestingly, some of the aptamers that showed high affinity in the AlphaScreen measurements seems to have mediocre KD values according to MST. Nevertheless, we were able to pinpoint some aptamers of high affinity. In our opinion, this controversy might have occurred due to the fundamental differences between the two characterization methods. Meanwhile, AlphaScreen determines the interaction between two molecules that are immobilized. MST measures this interaction in free solution. This highlights the importance of choosing various methods during the characterization of aptamers, as both of these methods provided important highlights and insights into the practical applicability of these aptamers. So next, we performed cell-based assays with these aptamers, and for that we chose the human A549 adenocarcinoma cell line. It is a suitable RSV infection model. It was derived from the epithelial lining of the lung. So first, to address the cytotoxicity of aptamers, we treated this A549 cell culture with polivizumab or positive control, the modified aptamers, and our non-modified aptamers that serve as negative controls. And after 48 hours of incubation, we measured the viability of the cells with a resizorin-based approach, and we found that none of these molecules proved to be cytotoxic to the aptamers, to the A549 cell line. In order to determine the half-maximal cytotoxic concentration, this cell line was also treated with a dilution range of these aptamers, and we found that even in the highest applied concentration, we didn't see a drop in cell viability. Therefore, these results show that the cytotoxicity of the aptamers are not expected to hinder the therapeutic application of the modified aptamers. Next, in order to assess the stability of these aptamers, we again treated the A549 cell line with these molecules and took samples from the cell supernatant and performed qPCR in order to quantify the amount left in the supernatant. The high stability of the modified aptamer is clearly shown here. At least a third of its original concentration was still present after 24 hours of incubation. Meanwhile, its non-modified counterpart had only a few percent left after 24 hours of incubation. Therefore, these aptamers are expected to withstand the prevailing conditions of the lung for many hours. In the in vitro virus neutralization studies, we chose a recombinant strain of RSV. This RSV contains a gene that codes for the green fluorescent protein. So if a cell is infected with this RSV strain, it will also express GFP, and the number of infected cells will be proportional to the fluorescence intensity. This RSV was pre-treated or pre-incubated with the molecules in question, or modified aptamers and polivizumab, and then the A549 cells were infected at a multiplicity of infection of 1. After removal of the unbound particle after 1 hour, we performed the total fluorescence measurement for 48 hours. Then these cells were subjected to fluorescence microscopy imaging, and finally they were lysed in order to quantify the viral genomes, the amount of viral genome by RT-qPCR. In the total fluorescence measurements, the lowest fluorescence was measured in case of the aptamer or polivizumab pre-treated RSV infected cells, showing that these molecules possess virus neutralizing effect. The highest signal was produced by the untreated RSV and our negative control aptamer pre-treated RSV infected cells as expected. This was further validated by the RT-qPCR of the viral genome, showing the same effect taking place. These aptamers possess a comparable antiviral effect to that of polivizumab. In the fluorescence microscopy of cells, we see that the untreated RSV infected cells and non-modified aptamer infected cells show the highest number of fluorescent cells, as well as syncytia formation. These large cells are the syncytia, which was clearly reduced in polivizumab or modified aptamer pretreated samples. In the dose-response analysis, we found that all of these modified aptamers in question possessed a dose-dependent virus neutralizing effect. We calculated the health maximal inhibitory concentration for polivizumab and our best candidate aptamer D4, which turned out to be around 5 nanomolar for polivizumab, which is in concert with the previously published values. And for D4, it was very close to this value. This also proves that these aptamers possess virus neutralizing effect, which was also validated with the qPCR of the viral genome. To summarize our results, we were able to generate a tryptophan-like side chain holding aptamer library by Kodak cell and water and oil emulsion PCR, which was then used in a ModSelex protocol to produce high affinity aptamers that can differentiate between the conformational variants of the F protein. These aptamers did not prove to be toxic in the applied cell culture, and they remained stable for quite a long time. And most importantly, in this cell culture, we could prove that these modified aptamers inhibit RSV infection with a comparable efficacy to the prophylactic drug used in clinical settings, polivizumab. We think that virus protein selective aptamers can yield efficient and cost-effective drug candidates, and we hope to see them in a clinical setting later. I would like to thank my supervisor, Tamás Mészáros, and the members of our research group for their continuous help and support, and our collaboration partners, Peter D. Kwong from the NIH, whose group supported us with the stabilized versions of the F proteins, and Marine de Jong, who provided me the opportunity to perform these virus neutralization studies at his lab. And I would also like to thank the audience for their kind attention.